A body lies on the sidewalk near an alley. It looks like the victim fought off her attacker, leaving blood and tissue under her fingernails. There is also a pool of blood on the sidewalk next to the victim. Two suspects have been picked up just a block away with fresh scratches on them. Could one of them be the killer? DNA fingerprinting can be used to determine if one of the suspect's DNA matches DNA found at the crime scene. The necessary blood samples from the crime scene, the victim, and the suspects can be collected and analyzed. Here are the results of the analysis. Let's compare the DNA fingerprint of each suspect to the blood samples we found at the crime scene to see if there are matches. We can also compare these results to the DNA fingerprint of the victim to make sure the crime scene sample isn't just from the victim herself. Suspect 2 may be a suspect, but this evidence would not hold up in court. There is only one band found in both his sample and the blood from the crime scene, the fourth band from the top in his DNA. However, the victim has this same band in her DNA. The band could have come from either the victim or the suspect. The DNA evidence of suspect 1 points us to the real murderer and makes a strong case for conviction. DNA fingerprinting. DNA fingerprinting is based on variations in the length of repetitive DNA sequence clusters called mini-satellites found throughout the genome of an individual. This video demonstrates how DNA fingerprints are generated and how they can be used in forensics and archaeology. Mini-satellites are clusters of nucleotides ranging from 15 to 100 base pairs in length. These clusters are present as strings of tandem repeats. The nucleotide sequence shown here has the base sequence GG, AA, G repeated 4 times. The number of repeats at a given chromosomal locus ranges from 2 to over 100. The loci are called variable number tandem repeats, or VNTRs. Each variation in the number of repeats represents a different allele. Mini-satellite regions sandwiched between 2 restriction sites, or between 2 known sequences for which PCR primers can be constructed, can serve as useful genetic markers. Here, each GG-AAG repeat is symbolized by a blue box. VNTR regions are cut from the chromosome with restriction enzymes, run on a gel, and visualized with southern blotting. The alleles with fewer repeats will move more quickly on the gel than the longer fragments with more repeats. A single VNTR site may have dozens of alleles, making it likely that an individual will have 2 different alleles for a given VNTR. The VNTR here, designated VNTR-A, comes in multiple alleles. One individual carries alleles A6 and A2, and another carries A3 and A4. When the DNA from these individuals is digested with restriction enzymes, electrophoresed on a gel, and southern blotted, the pattern of VNTR alleles can be seen. VNTR sequences from different loci can be combined to create a DNA fingerprint. In this example, a second VNTR, designated VNTR-B, is used in the fingerprint. The banding patterns that result from a DNA fingerprint are highly variable. Provided that enough VNTR sites are used, each individual's pattern is theoretically unique. Here, 2 VNTRs are sufficient to determine that the 2 samples are from different individuals. DNA fingerprinting forensics. DNA fingerprinting has a wide variety of modern applications, including comparison of genetic samples in criminal trials. A little over a dozen VNTR probes are routinely used in forensic analysis. In an individual sample, 4 to 10 probes are typically used. In the example here, 2 probes have been used. Assuming that we know the frequency with which each allele occurs in the U.S. population, we can calculate the probability that the evidence DNA has come from an individual other than the suspect. For now, let's focus on the alleles of VNTRA. The frequency of occurrence of the A6 allele is 1 in 20 individuals, so we would expect that 5% of the American population would have this allele. The frequency of the A2 allele is 1 in 12 individuals. What is the frequency of having both alleles in the same individual? The frequency of occurrence is equal to the product of the two individual frequencies. 1 over 20 times 1 over 12 equals 1 over 240. Only 1 in 240 individuals would show this pattern for VNTRA. Let's now also consider VNTRB, the pattern for which appears when we use the second probe. The frequency of the B2 allele in the population is 1 in 345, and the frequency of the B1 allele is 1 in 90. The probability of having all four alleles is determined by multiplying the frequencies of each allele. 1 in 7 million individuals are expected to have this combination of alleles. Statistically, about 37 individuals in the U.S. share this DNA fingerprint. Additional VNTR probes would increase our confidence that the two samples are from the same person. However, keep in mind that alleles are not truly randomly distributed in the population. Close relatives are more likely to share similar patterns. The ancient remains of several human bodies have been discovered and legally need to be returned to the tribe that contains the closest descendants. You have been called as a witness in the resulting legal case to give your opinion. Let's go over your testimony. After obtaining the samples, preserved bone and muscle fragments, you conducted a DNA extraction, which allowed you to get a relatively pure vial of DNA out of the sample. This and subsequent steps were done very carefully to avoid contamination. Because you suspected the ancient DNA would be degraded, you pre-treated it with random primers and the Klenow fragment, a procedure found to help repair damage and aid amplification, and amplified the DNA using polymerase chain reaction, which is a technique that allows you to rapidly create many copies of the DNA. Random priming will allow you to amplify all the DNA rather than a specific fragment. To make sure that the DNA you analyzed was indeed from the ancient sample, during these procedures you used both positive and negative controls to test the quality of the experiment. A positive control, such as a modern DNA sample, would verify that the technique is working. A negative control, for instance no DNA, would show a lack of contamination. Following amplification, you used restriction enzymes to cut out the DNA fragments with the genes you wanted to study. Restriction enzymes can be used to isolate the genes of interest, such as VNTRs. Then you used gel electrophoresis to separate the DNA fragments. During gel electrophoresis, smaller fragments will migrate faster than larger fragments. After separating the DNA fragments using gel electrophoresis, you performed a southern blot in which you transferred the DNA onto a filter and then incubated the filter with a radiolabeled DNA sequence complementary to the gene you were interested in. Southern blot is the standard procedure used to prepare DNA for probing. This final step with the radiolabeled DNA sequence is called hybridization to a probe. The probe hybridizes to the gene of interest and can be visualized by autoradiography for a radioactive probe. This step can be repeated for multiple genes. Then you used several highlight variable loci to create a specific DNA fingerprint, which is a possible unique banding pattern frequently used in forensics. Finally, you compared the sample to the modern DNA samples in order to determine the group most closely related to the individual from whom the sample was taken. To sum it up, mini-satellites are clusters of nucleotides ranging from 15 to 100 base pairs in length that are present as strings of tandem repeats. The number of repeats at a given chromosomal locus is highly variable, and so these loci are called variable number tandem repeats, VNTRs. Each variation in the number of repeats represents a different allele, and there may be dozens of alleles in a population, making it likely that individuals will be heterozygous for any given VNTR. VNTRs from different loci can be combined to create a DNA fingerprint, and if enough sites are used, each individual's pattern is theoretically unique. DNA fingerprinting can thus identify specific individuals in a large population. If the frequency of occurrence of each allele in the population is known, the frequency of several alleles occurring together can be calculated by multiplying the individual frequencies, provided that the loci are on separate chromosomes. However, care must be taken in interpreting DNA fingerprinting results because the alleles are not truly randomly distributed within a population, and close relatives are more likely to share similar patterns.
Gel electrophoresis of DNA. DNA molecules of different lengths can be separated by gel electrophoresis, and the lengths of the DNA molecules can then be determined. In gel electrophoresis, DNA molecules migrate to the positive pole as current passes through the gel, with the shortest DNA molecules moving the farthest. Making the gel is the first step in gel electrophoresis. A common material used for gel electrophoresis of DNA is agarose. Agarose gels are made by first boiling a mixture of powdered agarose and buffer. When the mixture has cooled to about 65 degrees Celsius, the solution is poured into a gel mold. When further cooled to room temperature, the agarose solidifies to produce the gel with indentations called wells. The agarose gel is placed in an electrophoresis apparatus and buffer is added to cover the gel. DNA samples are loaded into the wells using a micropipetter. One sample contains DNA molecules of known lengths so that the experimental DNA samples can be compared to it. Each DNA sample has tracking dyes added to it, usually blue, and either sucrose or glycerol to make the solution dense and sink into the well. After the wells are filled, the power supply is turned on and the DNA moves towards the positive pole. At this stage, the DNA band or bands can't be seen, but the tracking dye, dark blue, allows the progress of the electrophoresis to be followed. Smaller DNA fragments move faster down the gel than larger fragments. The DNA is visualized by staining, for example with ethidium bromide, and then destaining in water. The ethidium bromide binds to the DNA and makes it fluoresce orange-pink under UV light. A photograph of the results is taken and used for analysis and for a permanent record of the experiment. The lane on the right has the DNA molecules whose lengths are known. The lengths of these size standards are given as the number of DNA base pairs, BP. The longest DNA molecules move the slowest, so they're at the top. They have 23,130 base pairs. Moving down, the DNA molecules in the bands have 9,416 base pairs, 6,557 base pairs, 4,361 base pairs, 2,322 base pairs, 2,027 base pairs, and finally 564 base pairs for the short, fast-moving DNA molecules. To determine the sizes of the DNA molecules whose lengths are not known, their positions are compared to the DNA molecules of known lengths. For the sample on the left, the DNA molecules are 6,201 base pairs and 1,268 base pairs. For the sample in the middle, the DNA molecules are 4,796 base pairs, 1,405 base pairs, and 1,268 base pairs. Notice that the shortest DNA fragments in the left and middle samples are the same lengths, indicating that these DNA samples were cut at the same sites, the fragment's ends, with the restriction enzyme that was used.
Polymerase chain reaction. The polymerase chain reaction, PCR, is a rapid, cell-free method of DNA cloning that has extended the power of recombinant DNA research. This video presents the steps involved in PCR and demonstrates how the technique can exponentially amplify specific DNA sequences. Polymerase chain reaction, PCR. There are 3 steps in the PCR process. 1. Denaturation of the source DNA. 2. Annealing of primers. 3. Extension of primers. In the first step of PCR, the double-stranded DNA in the sample is denatured into single strands by heating at 90 to 95 degrees Celsius for about 5 minutes. This temperature is critical. At a lower temperature, the DNA might not fully separate, but at a higher temperature, some components of the reaction might degrade. In the second step, the reaction temperature is lowered to between 50 degrees Celsius and 70 degrees Celsius. At this temperature, primers can bind to the denatured DNA. These primers are synthetic oligonucleotides, 15 to 30 nucleotides long, that bind specifically to sequences flanking the segment to be amplified. The primers serve as the starting points for the synthesis of new DNA strands complementary to the target DNA. Custom-made primers are commercially available. In the third step, a DNA polymerase extends the primers by adding nucleotides in the 5' to 3' direction, making a double-stranded copy of the target DNA. DNA synthesis is performed at temperatures between 70 degrees Celsius and 75 degrees Celsius. The polymerase is a heat-stable form called Taq polymerase, derived from Thermus aquaticus, a bacterium that lives in hot springs and has evolved temperature-resistant enzymes. A less thermostable enzyme would degrade when subjected to the relatively high temperatures required to denature DNA. This extension step represents the end of a PCR cycle. The steps of the cycle are repeated, this time beginning with 2 DNA molecules. After 2 cycles, there are 4 DNA molecules. With each additional cycle, the number of copies theoretically doubles, so there would be 8 copies after 3 cycles and 16 copies after 4 cycles. It only takes about 20 cycles to increase the number of DNA copies by 1 million. Let's watch this virtual PCR lab's reaction progress during the incubation process. Note that the upper diagram shows the temperature changes in the reaction tube for each cycle. The graph below plots the number of DNA copies per reaction cycle. Assuming we started with 1 DNA molecule, we will have 2 molecules after the first cycle of PCR. After the second cycle of PCR, we have 2 times 2 equals 4 copies. After the third cycle, we have 4 times 2 equals 8 copies. Note that we can calculate the number of copies as 2 times 2 times 2 equals 2 cubed copies. The theoretical number of copies is 2 to the power of n, where n is the number of cycles. After 4 cycles, we have 2 to the power of 4 equals 16 copies. After 5 cycles, we have 2 to the power of 5 equals 32 copies. So far, the reaction has been running about 40 minutes. Now, after 10 cycles, there are 2 to the power of 10 equals 1,024 copies. On the lower graph, notice how the scale on the y-axis has changed. After 15 cycles, there are 2 to the power of 15 equals 36,768 copies. On the lower graph, notice how the scale on the y-axis has changed again. At the end of 25 cycles, theoretically there are over 30 million copies. In practice, the reaction rate may decrease in later cycles. If a larger number of copies were needed, it would eventually be necessary to set up a fresh reaction. On the lower graph, notice how the scale on the y-axis has changed again. PCR allows rapid direct amplification of specific target sequences of DNA that initially may be present in infinitesimally small quantities. The 3 steps in a PCR cycle are denaturation, annealing, and extension. DNA molecules are first denatured by heating at 90 to 95 degrees Celsius to dissociate the double-stranded molecules into single strands. During annealing, synthetic primers, oligonucleotides, 15 to 30 nucleotides long, bind specifically to sequences flanking the segment to be amplified. These primers act as starting points for the synthesis of new DNA strands complementary to the target DNA. During extension, a heat-stable form of DNA polymerase, such as Taq polymerase, extends the primers by adding nucleotides in the 5' to 3' direction, making a double-stranded copy of the target DNA. Each PCR cycle theoretically doubles the number of copies of the target molecule, such that 25 cycles may result in more than a million-fold increase in the number of DNA copies. Overview. Restriction fragment length polymorphisms, or RFLPs, are variations in DNA fragment length generated by restriction enzymes. This video describes how RFLPs are detected and examines how analysis of their inheritance patterns can reveal markers for genetic traits and disorders. Restriction fragment length polymorphisms. The DNA fragments shown represent different alleles from homologous chromosomes, labeled A and B. The region of chromosomal DNA that would hybridize to a particular probe is shown in green. The arrows represent sites where a particular restriction enzyme will cut the DNA. On chromosome A, 3 restriction sites create 3 kb and 7 kb fragments. On chromosome B, only 2 restriction sites are present, producing a 10 kb fragment. The absence of the middle restriction site in B could be due to a single base mutation. An experiment is performed in which genomic DNA is digested with the restriction enzyme, and then the digested DNA is electrophoresed on a gel, transferred to a DNA binding filter, and hybridized with the probe. The differences in restriction sites are inherited in a codominant fashion, so 3 different chromosomal genotype patterns can be recognized: homozygous AA, heterozygous AB, and homozygous BB. The differences in the patterns of fragments are referred to as restriction fragment length polymorphisms. RFLP analysis and inheritance. DNA samples from multi-generational families, 3 or more generations, can be used to localize a genetic disorder on a chromosome. The family represented here has members who are affected by a dominant trait, as indicated by the orange symbols. To identify the chromosomal locus of a genetic disorder, it is important to identify an RFLP DNA marker that co-segregates with the disorder. The marker exists in most individuals with the disorder, but not in most individuals lacking the disorder. Selecting an informative marker is largely a matter of trial and error. The most useful RFLPs are those for which most members of the family are heterozygous, so that each member of a chromosome pair can be identified. Members of this family carry 2 alleles for a restriction site. The A allele has a 5.0 kb fragment when digested with the enzyme. The B allele is composed of 2 fragments, one of 2 kb and the other of 3 kb. In this experiment, genomic DNA from individual family members is digested with the restriction enzyme. The digested DNA is electrophoresed on a gel and then transferred to a DNA filter for southern blotting with a probe that hybridizes to the DNA indicated by the green color. If the locus of the RFLP and the locus of the disorder are near each other on the same chromosome, they will show linkage. Although most RFLPs tested will not show linkage to the disorder, the RFLP in our example does show linkage. In all cases, individuals with the disorder carry the RFLP allele shown on chromosome V. On the other hand, most of the family members without the disorder lack this RFLP allele. Female 5 in generation 2, 115, married into the family. She does not have the disorder but does carry the RFLP allele we associate with the disorder. In this case, we presume that one of her chromosomes underwent crossing over in previous generations to yield this unique combination of alleles, a normal gene combined with an RFLP allele we associate with the disorder. The more closely two loci are linked, the lower the probability of recombination. Individual 3-1 probably received this recombined chromosome with a normal gene for the disorder from her mother and the RFLP allele from chromosome A from her father. So she is heterozygous for the RFLP but does not have the disorder. Commonly, RFLPs are used to identify potential carriers of genes for human diseases, but many other applications are possible. Suppose you would like to optimize a dog breeding program to select for blue-spotted dogs, resulting from a single mutation rather than black-spotted dogs. You have collected a set of DNA samples from a multi-generational family of dogs. You have also identified 3 possible RFLPs which might serve as useful markers for the mutation. The dog individuals colored blue in the pedigree chart all have blue spots. The 3 rectangles below the pedigree show different gels and bands associated with 3 different RFLP groupings. Which RFLP grouping would be the best marker for the blue-spotted phenotype? While trying to identify the best marker for the blue-spotted phenotype, you need to remember 2 small things. The blue spots are a dominant trait, and, ideally, all the blue dogs would share at least one band. You also need to think whether all these alleles are correlated. This will definitely lead to the conclusion that all of the blue-spotted dogs contain the lower band and can be a useful marker for the trait. Sickle cell anemia is one of several known genetic disorders which results from a mutation in the beta-globin gene. This autosomal recessive condition is caused by a single nucleotide change from GAG to GTG. This difference also results in a characteristic RFLP pattern. Let's explore the relationship between genotype and DNA migratory behavior in an electrophoretic gel. First, let's match the genotype with the RFLP patterns in the gel. We use the diagram showing restriction sites in the globin gene alleles as a reference. Now that we've matched the genotypes to the RFLP patterns, let's try to understand the pedigree chart based on our knowledge of sickle cell genetics. Note that the female have 1 copy of each allele, so she would not exhibit symptoms of sickle cell anemia, as it is a recessive trait. Among the children, the male is homozygous for the normal allele. He does not carry the sickle cell allele. The female child is the only member of the family who would show symptoms of sickle cell anemia, and the fetus is a carrier of the sickle cell allele, as are the parents. RFLPs are variations in the pattern of restriction fragments produced in DNA from different individuals. The fragments vary in length because of sequence differences that may result in the creation or removal of recognition sites for restriction enzymes. The patterns can be analyzed by southern blotting of the DNA fragments. If an RFLP is close enough to a disease-related gene and co-segregates with the disorder in a multi-generational family, it can be used as an informative marker for the disease. Because RFLPs are inherited codominantly, they can also be used to identify probable carriers of a genetic disorder. Analysis of human pedigrees. Analysis of pedigrees or family trees is useful in evaluating the genetic basis of phenotypic traits in organisms where controlled experiments to generate large numbers of offspring are not practical or ethically possible. Pedigree, inherited cataract. The hypothetical pedigree considered here is for eye cataracts in humans, an autosomal dominant trait. Generations in pedigrees are indicated by Roman numerals at the left of the diagram. Circles designate females and squares males. Parents are joined by a horizontal line, with a vertical line leading to their children. The individual whose phenotype was first brought to the attention of a geneticist or medical researcher is called the proband. Individuals known to express the phenotype have colored symbols. Individuals who do not express the trait have gray symbols. In this example, the proband is heterozygous for the trait and her mate is homozygous for the normal allele. In this model, one of their two children, a son, is also heterozygous for the trait, as are half of his children with his homozygous normal mate. Note that neither of the proband's parents had cataracts. This suggests that the mutation occurred during gametogenesis in one of her parents. This chart is an example of a pedigree for a single character. Notice how the genotypes of the progeny in each generation can be determined by examining the possible genotypes of the parents. Pedigree for color blindness. Inheritance of some traits is linked to transmission of the sex chromosomes. In humans, the Y chromosome has relatively few genes, primarily those associated with sex determination and secondary male characteristics. The human X chromosome has a much larger number of genes associated with a variety of physiological processes. X-linked refers to a condition caused by an allele of a single gene located on the X chromosome. In this pedigree, the female in generation 1 is colorblind. Because this particular condition is caused by a recessive allele, she must be homozygous for the allele. Note that the condition appears only in her sons and grandsons, but not in any of her daughters or granddaughters. This is a characteristic of rare X-linked recessive traits in humans and other species where males are the heterogametic sex. This partial pedigree of hemophilia indicates how the trait descended from Queen Victoria in the British royal family. Historical research has concluded that X-linked hemophilia was not present in Queen Victoria's ancestral lines, but careful consideration of the pedigree leaves little doubt that she was heterozygous for the trait. Pedigree analysis provides a method for analyzing inheritance patterns of human traits and disorders over several generations. Researchers constructing pedigrees for genetic analysis use a standard set of symbols and terminology. These analyses can be used to distinguish between dominant and recessive traits and to determine if traits are sex-linked. They can also be used to predict the probabilities of offspring with particular genotypes and phenotypes from individuals in the pedigree who have not yet borne children. Pedigree analyses can also identify the probable source of a mutation for traits that appear in a family without a prior family history of the trait. Probability. Mendelian genetics deals with the prediction of the genotypic and/or phenotypic ratios of various crosses. These genetic ratios are best expressed as probabilities. This video demonstrates how the principles of probability are used to predict and interpret the outcomes of genetic crosses. If you toss a coin, what outcome do you expect? You will, of course, see either a head or a tail. There are only two possible outcomes. If you toss the coin again, you will likewise get either a head or a tail, and the result of the second toss will not depend on the result of the first toss. What happens if you toss the coin many times? For a very large number of tosses, you should get heads about the same number of times as you get tails. With a large number of tosses, the probability of getting heads in any one toss is 50%, or half, as is the probability of getting tails. Let us take a penny and a dime and toss the coins simultaneously and consider the results. Notice that the outcome for the penny does not affect the outcome for the dime and vice versa. When you are tossing the penny and dime simultaneously, there are 4 possible outcomes. A. Penny head, dime head. B. Penny head, dime tail. C. Penny tail, dime head. D. Penny tail, dime tail. As the total number of tosses increases, the frequency of each outcome approaches the predicted probability of 1/4. These results illustrate the product law of independent probabilities. The probability of obtaining a head or tail for either coin in a given toss is 1/2. The probability of getting any one of the possible combinations is 1/2 times 1/2 equals 1/4. Now, let us try to understand the probability of tossing a penny and a dime and obtaining 1 head and 1 tail. Let us now consider whether the penny or the dime comes up heads, provided that the other coin comes up tails. When you are tossing the penny and dime simultaneously, there are 4 possible combinations. A. Penny head, dime head. B. Penny head, dime tail. C. Penny tail, dime head. D. Penny tail, dime tail. The question we are asking is, what is the combined probability of cases where we have a coin showing head and another coin showing tail? Since a single toss of the 2 coins has a probability of 1/4 of resulting in combination of a penny head and a dime tail, and a probability of 1/4 of resulting in combination of a penny tail and a dime head, we can apply the sum law to determine the combined probability for these cases. 1/4 plus 1/4 equals 1/2. Binomial theorem. If A and B are the probabilities of the 2 possible outcomes of an event, then for any single event, A plus B equals 1. If each event is independent of the others, then the product law of probability tells us that for n events, A plus B to the power of n equals 1. For any integer value of n, the binomial theorem lets us quickly write the expansion of the left side of this expression. Individual terms in the expression tell us the probabilities for different combinations of the A and B outcomes. Expansion. The binomial theorem provides the general expansion for A plus B to the power of n, where n is any integer, but A and B can be any number. For small values of n, we can easily determine the expansion of A plus B to the power of n by performing the multiplications explicitly, as done at left. In fact, we can always perform such multiplications to evaluate A plus B to the power of n for any integer value of n. But except for very small values of n, the multiplication can be tedious. Fortunately, the binomial theorem provides a labor-saving shortcut. Pascal's triangle. The binomial theorem tells us that the general expansion of A plus B to the power of n contains n plus 1 terms, as shown at left. The coefficients of the terms in the expansion can be read from a construct known as Pascal's triangle. For a given value of n, the leftmost number in a row of the triangle is the value of the first coefficient in the expansion. The next number in the row is the next coefficient, etc. Note that in this triangle, each number is the sum of the 2 numbers immediately above it. The coefficients can also be calculated from a formula. Occurrences. Now, suppose we want to know the probability of k occurrences of the event with probability A and m occurrences of the event with probability B, where k plus m equals n. We need only calculate the value of the term in the binomial expansion that has the factors a to the power of k times b to the power of m. Note that when the binomial theorem is applied to probability problems, the coefficients in the expansion indicate the application of the probability sum rule. For example, the coefficient of a to the power of k times b to the power of m tells us how many different ways the event with probability A can occur k times, and the event with probability B can occur m times. The binomial theorem can be used to calculate probabilities in cases where one of two outcomes is possible during each of a number of trials. For example, you might wish to calculate the probability that a couple planning a family of 4 children will have 2 daughters and 2 sons. So the total number of events will be 4, and the event A, or number of sons, will be 2, and the event B, or number of daughters, will be 2. Calculating, we find that the probability of having 2 daughters and 2 sons is 0.375, or 6/16. To sum it up, the laws of probability can be used to interpret the outcomes of genetic crosses. The product law states that the probability of 2 or more independent events occurring simultaneously is equal to the product of their individual probabilities. The sum law is used to calculate the probability of a generalized outcome that can be accomplished in more than one way. The binomial theorem is a mathematical result that provides the general expansion for the binomial a plus b to the power of n, where n is any integer. If a and b represent the probabilities of 2 possible mutually exclusive outcomes of a single event, and n is the total number of events, the theorem can be used to calculate the probability of any combination of a and b outcomes.